Papa. Horacio, welcome to our session. I will just give you a brief introduction to Horacio. He has been working with us at USP since 2003, and he is the Director of General Chapters Department Science Division. Um, he is providing us the scientific leadership to a team of scientific liaisons responsible for the activities of 6 different expert committees that cover the majority of USP General Chapters. Horacio earned his PhD in pharmaceutical chemistry from the University of Buenos Aires. He has authored many publications and peer-reviewed articles and is a frequent speaker and instructor on topics related to chromatography and validation. Prior to joining USP, he worked in the pharmaceutical industry in QA and QC. Horacio held the position of assistant professor of quality control in the Faculty of Pharmacy at Buenos Aires University and executive secretary of the Argentine Pharmacopoeia in the period of 1997 to 2001. He's a quality engineer certified by the American Society for Quality. So welcome, Horacio. To our participants, if you have questions, we do have the Q&A box available for you. It should be popping up on your lower right corner. So please type in your questions if you have any while Horacio is presenting. And we ask you that you please type out your questions fully and legibly so we really understand what the question is that you're asking, because we won't have an opportunity to really ask you back to help clarify. Our webinar will be for 1 hour. We will have a Q&A session towards the end, around 15 minutes before we are finished. And there will also be an exit survey for you. So we ask you to please give us your feedback. It really helps us to help improve as well on your experience today with USP. All right, with that, well, thank you so much. Please enjoy, Horacio. The floor is all yours.
Thank you, Ian. Good morning, good afternoon, good evening. Thank you for joining us today. As Ian explained, my presentation today is about a new general chapter we are planning to incorporate into the USP. Is chapter 1220. And my intention here is to explain, obviously, what is inside that chapter, analytical precision life cycle, and also explain what is the status of or what will be the status of this chapter in the context of USP, especially considering that we have a series of chapters that talks about validation, verification, and transfer, 1224, 1225, and 1226. So, if you look at the definition of validation, I picked the one in our USP chapter 1225, but the others definition are very similar. And it says that validation of analytical procedure is the process by which it is established by laboratory studies that the performance characteristics of the product meet the requirements for the intended analytical application. So the concept is that through the validation experiments, you will determine if the method has the appropriate variability in order to be used for one particular purpose. And we know that that is not always the case in the pharmaceutical industry. In many instances, the validation exercise is made in a checkbox type, and there is no clear connection between the variability of the method and the requirements for the particular analytical application. 1220 had the intention to solve that, or at least to provide a path that allowed us to develop methods that that will meet the requirements of certain predetermined requirements through the life cycle of the method. So from the moment that the method is implemented in the lab until the method is retired. In the introduction, 1220 says that this chapter presents an alternative framework for analytical procedures that holistically incorporates all the events that take place over the procedure life cycle that are designed to demonstrate that the procedure is and remains fit for the intended purpose. And I want to make clear that the intention here is not to replace 1224, 1225, 1226 with 1220. As the introduction says, 1220 present an alternative framework. So our expectation is that some people may use this enhanced approach for development and validation of their methods. Others may continue doing the traditional. And I think that there will be a transition probably that people will use one or the other. And the expectation is in the future, 1220 will be the norm. So you probably are familiar with this table. This is the table in the chapter on validation in USP, but it's a very similar table is in the ICHQ2 guidance. And as you can see, the table identify what are the experiments that you need to perform to determine the appropriate performance characteristics, depending on what is the intended purpose of the method. Being category 1, the main component within the matrix, category 2, a minor component in the matrix, category 3, is a performance testing, and category 4, an identification test. But and I will say this again, but with this series of experiments, you cannot determine if the variability of the method can be suitable for the intended purpose. As you know, we always say that you can validate any method, a very crappy method or a very good method. All of them can be validated. So again, we are trying to cover that gap in determine if for that particular application, the method or the variability of the method is suitable. FDA identified this problem. There's a document in 2004 where FDA talks about variability or unknown variability of the methods. The document says variability and or uncertainty is a measure in a measurement system can pose significant challenge when out of specification results are observed. So FDA understand that when you have an out of specification result, it's difficult to identify if it's because the variability of the method or is because what we are testing is outside the specifications. And this is because probably the total variability of the method had not been studied before. Measurement system variability can be a significant part of the total variability. And when similar and repeating out of specification observation for different products across the industry and less than optimal understanding of variability, this is what they are seeing, at least at that time. And in this context, it's difficult to promote the continuous improvement. So again, the industry is in many cases using ICHQ2 or 1225 to validate the methods. In many cases, trying to fulfill a regulatory requirement as a checkbox exercise, as I explained before. But again, the connection between the reportable result and the predefined requirements for the method are not perfectly identified. There is no such a link between these two things. And so this is basically one of the concepts we identify when we propose the modification of the current validation of analytical methods. That started in 2013, I think. USP created an expert panel with the intention of developing an approach for validation of analytical procedures, but we're looking into the future. So our question to the panel was, what do you think validation will be done in the industry between 20 years from now? The panel very quickly identify the approach that is more or less identified in Q8 and now Q12 as an approach that can be applied to analytical procedures. Obviously, this is not a finding from the expert panel. Is not a something that they decided to do. There were many groups at that time working in this concept, so they basically embraced that concept. Following that, there was a series of 4 papers that the expert panel published. The first one was basically a philosophical definition of the life cycle management of analytical procedures. So this is where the basis of the concept were explained and developed. And follow that, there were 3 papers that expand on very specific topics within the analytical procedure life cycle, especially those that are kind of a new for the industry, in order to better understand and apply the life cycle concept. So as you can see, there was a paper on fitness for use, decision rules, and target measurement uncertainty, MPF 42.2. There is one on analytical target profile, structure and application through the analytical life cycle, and a paper on analytical control strategy. All these elements are part of the life cycle approach for the method. And what you see on the other side of the slide is basically the scheme of the process. And as you can see, everything starts with the quality product target profile, which is defined in QA. So knowing what we want to measure, we can evaluate the amount of error we can have in our method in order to not avoid making the wrong decision when we approve a product. So, and we can quantify those requirements in form of total error, uncertainty, wherever you prefer, but that the analytical target profile is simply a very simple declaration of what is the intended purpose of the method and what is the maximum error I can accept in this measurement according to the criticality of the quality attribute we are measuring. The process of the life cycle is divided in 3 stages: procedure design, procedure performance qualification, and ongoing procedure performance verification. You can see the word validation is not here. And this is important to mention. In this concept, validation takes a more important role, which is overarching the whole life cycle of the method. So I would say you will develop the method, you will validate the method, and you have to demonstrate that this state of validation is maintained through the life cycle. The actual validation of the method is stage 2, procedure performance qualification. As we will see later, is quite of a, it may be a little different from the classical Q2 validation. So there is another important component in here, which is knowledge management. There is a recommendation for a very strong process of gathering information about the method that will be used during the procedure design. Also, as the method advances in the life cycle, we will be able to produce or data that support the validation, and that also will be knowledge that will be incorporated into the body of knowledge we have from the for that particular method. So again, stage one is properly the method development, is when we decide we design the method, and we establish the necessary parameters for the method. And it's very important here that we will identify or better understand the effect of the procedure parameters on the performance of the method. We will do a risk assessment, and we will identify again some kind of is a robustness study, but we will identify a space where the method can be used. On stage two, we basically will confirm that the reportable value generated by the procedure meet the ATP criteria. So once the confirmation, it is confirmed that the method meet the requirements of the analytical target profile, the method can be implemented. And after that, the purpose is that you have to collect data regarding the performance of the method in routine use, to identify in advance any problem that the method may have. And if the problem happens, you have to make the decision of going back to stage two or going back to stage one to fix the problem, depending on the criticality of the problem. So I mentioned a couple of elements that are very inherent to the life cycle approach. One of those is the analytical target profile, or ATP. And we say that ATP is a prospective description of the desired performance of an analytical procedure that is used to measure a quality attribute. So technically speaking, we will have an ATP for one particular procedure, for one particular attribute we want to measure. This ATP, or this description, is established before the method is developed, because the definition of what is the appropriate quality of the reportable value had to be done before selecting the method. And based on that, we can choose the proper method technology for the measurement. It says that the ATP is independent of the measurement technology. However, and recently we hear more and more that maybe is not the case all the time. There are cases that the ATP needs to be associated with one particular technology. So I think that many of these concepts are evolving as we speak. So we can expect that 1220 in the future may be revised to incorporate new ideas or new concepts, or modify some of the elements described there. What should include an ATP? As a minimum, it is important to define what is the analyte, so what we are trying to measure. A brief description of the analytical matrix. So we will measure A in the presence of A, B, C, D. Also, we will describe the range that we will use the method. Remember, this is based on the specification of the quality attribute we want to measure. And finally, the precision and accuracy acceptable for the reportable value. This is very, very important. And as you can see through this presentation, the core of validation is now precision and accuracy. All the other performance characteristics are, I would say, secondary. The main aspect is that if you meet the precision and the accuracy established for the method within the range, technically speaking, all the other components or all the other performance characteristics will be in the range we specify. The chapter explain or present a couple of examples on ATPs. Again, this is based on the current knowledge. The expectation is that this will be evolving. In fact, in the process of elaborating this document, we search in the literature for how different authors are using the term analytical target profile, and we found at that time, which is 2 or 3 years ago, almost 30 different ways that the ATP is presented. Anyway, the intention here is to be very flexible in terms of what you can include in the ATP. However, you have to keep in mind that the elements we mentioned before needs to be there. In these two examples, the ATP is presented with different level of complexity, if you want. The first example, as you can see, we identify what is the analyte, we identify the range, we identify what is the matrix, the analytical matrix, and then we identify the maximum accuracy and the maximum precision. This is kind of based on a classical validation mode where accuracy and precision are estimated as two separate performance characteristics. We are now advocating for estimating the total analytical error in the method as the sum of the accuracy and the precision. And this is where the method, the example 2 is doing, when it says that the distribution of the total analytical error of the reportable value falls within the total allowable analytical error range of plus minus something. So again, in this case, the ATP defined the maximum variability for the method, and this is what you want to try to achieve during the method development and confirm during the stage 2, the qualification and the continuous verification. As I mentioned before, precision and accuracy take a very important role in the process of defining the total analytical error. We use bias, the term bias in the chapter many times. Maybe this will create confusion in the future because you know that the accuracy definition in ISO is different to the accuracy definition in ICH, and all our documents are based on ICH. But we thought that at some point is important to try to align to a better terminology. So bias is the way that ISO define what we define as accuracy in ICH. So again, precision and bias or accuracy will give me the variability, the total variability of the method. And we have to identify if that variability is appropriate for what we are trying to measure. So for example, if you have a maximum, the specification based on a typical 90%, 110%, you don't want to have a method that when you add the accuracy and the precision, most of that range is covered by the distribution of the test results. You want to have, and this is like a rule of thumb, you want to have that the total variability of the method cover at least 1/3 of the budget for the acceptance criteria, and no more. So how we will identify what is the appropriate variability? Well, we have to use different factors. We have to take into account how critical is the quality attribute that we are planning to measure. What is the risk of making the wrong decision? So approving a product which is not good, or rejecting a product that is good. And the width of the specification of the acceptance range for the quality attribute measured by the procedure is the other aspect you have to have. You know, if suppose we are talking about an API where the maximum, the acceptance criteria is 98,102, is the method may be different from one that we want to use to release a product with a 90% 110 acceptance criteria. And finally, the clinical safety and efficacy impact that the analytical error may have, especially in those products that has a very narrow clinical window between the toxicity and the effectivity. This is where this approach makes much more sense in order to better identify the accuracy and the precision as a total analytical error. This try to explain a little bit the different situations that you may encounter. So suppose that this is the upper limit of an specification. And you see there are 4 reportable values, and the identification of the distribution of those reportable values. And as you can see, in the case of scenario 1 and scenario 4, the decision is very clear. In one case, you have the complete distribution of error outside the limit, and in the other case, in scenario 4, you have the whole distribution of the error inside the specification. The problem is when you are in a scenario 2 or 3, where a certain amount of the results will be inside, but the majority outside, and the opposite. Only the tail will be outside, and the majority, the rest of the distribution will be inside. So what we are trying to identify is how much of this type of error we can accept in making a decision about a result. The decision rules that we use typically for established specifications take this into account, even though it's not specifically identified as a value. There is we can assume that the analytical error will be at least incorporated if we use the appropriate decision rule. For example, if you look at the first one, this is the typical case where what the acceptance zone is more narrow than the safe and efficacious range. So you have a guard band that will allows you to be safe in the decision about approving or not. So if you are a little bit outside the method in both of the limits, the guard band will give you the assurance that the method is still within the safe and efficacious range. There are other cases like the second figure that you can use when you need to reduce the risk more, and you establish a strict acceptance zone, as we say before, between the values that are narrow than the safe and efficacious range, and you add in each case an indecision zone. So there is a zone inside or out, yeah, and outside, that it will not give you the appropriate assurance that you are within the range that you need. So in the decision, if you get a result within the indecision zone, will be to take another sample and make additional testing. This is familiar for you because it's more or less the scheme used in tests like dissolution or content uniformity. So going back to the different stages, trying to put a little more information on each, what happened in each case. So stage one is a procedure design. So we will collect knowledge. We will do experiment. We will do risk assessment in order to identify what is the appropriate method. Remember that at this point, the ATP is already defined. So we will develop a method that we are sure that the reportable result will meet the criteria established in the ATP. Minimize the bias and provide the adequate precision. So again, this is not about minimizing the bias and minimizing the precision. The precision, it may have an appropriate value depending on the ATP requirements. Also, we need to understand the effect of the procedure parameters in the performance of the method. In fact, we use the robustness studies to do this. However, this approach advocates for more in-depth investigation about the control, the effect of the procedure parameters using design of experiments and defining what is called method operated design region, MODR, which is a space where the method was designed to be to be sure that we are meeting the requirements of the ATP. Based on that, we have to optimize the performance characteristics of the analytical procedure, including accuracy, precision. We will have to describe the calibration model. We have to determine the selectivity and the sensitivity of the method, etc. The other two components that are added here is the definition of a preliminary replication strategy. So the understanding is that in order to have a reportable value that can be compared with the acceptance criteria, we have to have certain number of results in order to be sure that the precision is adequate. We also will define an analytical control strategy. As we mentioned before, on stage 3, you will start monitoring the method during the rest of the life cycle. And in monitoring the method, you have to choose what are the appropriate parameters and activities you need to do to demonstrate that the method is still meeting the requirements in the ATP. System suitability is part of it, but we will realize that there are others that can other quality tools that can be used to demonstrate the continued performance of the method. So in order to measuring the risk, because the risk assessment is also part of stage 1, again, we can use any of those instruments that are used to support the quality management, like any quality tool. In the chapter, we describe the use of two, the Ishikawa diagram and the heat map that I will show in the next one. So the idea here is based on the results we obtained during the robustness study, or MODR, we can identify what are the critical parameters in the performance of the method. And for that identification, we will use the Ishikawa diagram. Then we have to determine the criticality of that parameter we identify. And for that, we can use a heat map, like in this case. So identifying the parameters or part of the method that can be critical for the result. You can identify how much this will impact the level into strong, medium, and minor. And the impact is, in this case, determined based on the accuracy and the precision. During stage 2, again, this is where we confirm that the method was developed to meet the ATP requirements, and also we will confirm the initial analytical control strategy that was identified as preliminary in the stage 1. One important thing is, again, the qualification will be based on the accuracy and precision. However, if there is a requirement, a regulatory requirement to complete all the other analytical performance requirements of the method, you can use data generated in stage 1 if needed. So as you can see, the collection of information we have in stage 1 and pre stage 1 and ATP, it can be used to present to the regulatory authorities for them to better understand the good or bad of the method. So at the end of the stage 2, the replication strategy is confirmed, and it's confirmed also that the performance of the procedure meet the ATP and other requirements. And finally, we enter in stage 3. Stage 3, again, is the monitoring of the method during the life cycle. And the idea here is to have an early indication of potential performance problems or adverse trends, and identify what is the changes required in the analytical procedure or in the performance of the method. Confirmation of procedure performance after changes, change management. It will be also guaranteed by the data of the continuous verification. Remember that when you detect an out of trend result, you may need to modify the method, and depending the level of the change, you may need to go back to stage 2, stage 1, and this have to be determined based on the risk management. One of the tools, again, quality tools here, one of the tools that you can use is control charts. In this example, which is in the chapter, is a range control chart, but you can identify data, in this case, out of specification. But in other cases, you can see clearly that there are 6 or more results in the same trend, and that may be an indication that that there is a need for the revision of the method. Finally, I want to spend a couple of minutes on the ICH activities, because in November 2018, it was approved a plan for the revision of ICHQ2. Again, the ICHQ2 was developed 30 years ago, and the technology changed, the need for better data change, etc. So it's clear that a revision of ICHQ2 is important. Again, that was in November 2018. The ICH Assembly approved the work plan for ICHQ2. In the first meeting, it was clear that instead of incorporating all these elements into one guidance, there is an opportunity to develop a new guidance. So in this particular case, the plan now is to revise ICHQ2 and create ICHQ14, which is called Analytical Procedure Development. So regarding ICHU2, the concept paper indicates that the current ICHU2 is very chromatographic oriented, and there is a need to incorporate the use of spectroscopic or spectrometry methods and multivariant methods that have to be evaluated using a different model, different approaches. So the typical ICHU2 parameters may not be appropriate. It also indicates that because there is no reference standard used during the analysis for multivariate methods, it is important that the robust of the method will be well identified, and the variability of the method will properly addressed. In terms of what is in the guide right now, the guide is still not in the public domain. Hopefully will be available between July and the end of this year. But the major changes are obviously the incorporation of the multivariate calibration methods, accuracy and precision are included in a, I would say, in a higher category. It's like there are the high category of performance characteristics, accuracy and precision, and all the others. So linear calibration mode, not linear calibration mode. We're trying to avoid the word linearity because in fact, it's not just such a thing. There is also an incorporation of the term selectivity, because the parameter what we call today specificity in chemistry is to demonstrate that the method is specific for something. However, what we do during the validation is to qualify the level of selectivity that the method has. You don't always need a very only a specific method. Q14 again is intended to be used during the method development. Incorporate some of the components that we described today. Important is that the method describes two possible approaches for method development: the minimal approach or the classical approach, and the enhanced approach, which incorporate the elements that we described in 1220, such as things like a analytical target profile, knowledge management, risk management. Obviously, the description of robustness will be now in Q14 instead of Q2. And then two important things: how to develop multivariate analytical procedures and real-time release testing. So again, these two guidance probably will be available before the end of the year. The group will meet again the first week of June to do the final adjustments before the publication. With that, I think that this is my last slide. Thank you very much for your attention, and I think that we open the floor for questions.
Thank you very much, Horatio. There are indeed a few questions. First question is, well, we have the normal ones about if we can provide the slides to these presentations, and also if we can share the recording. So we will share the slides, of course. I will send an email to all participants today or tomorrow with the PDF of the slides. That's completely possible. We will also share the recording on our YouTube EMEA webinar channel, and we'll let you know the link then as well once it's done. That will take, however, a little while, and will be done maybe in the next week. We have, of course, also a little bit of technical questions here. The first question that I would like to ask you, Horatio, there is some difficulty with the term multivariate analysis. Can you explain what you mean with multivariate analysis, please?
Yeah. So in the typical analysis, chromatographic, like you use like a two dimensions. when you go into the area of, especially in spectroscopic techniques and near, like a near infrared, for example, you will be determines. I mean, you have to develop a model that take into account more than two variables of the method, and that is the way that you will identify a result. It's not the classical chromatographic, but it's a multidimensional space where the result is incorporated. Basically, is that so it's very specific for certain spectroscopy test, but it's using more and more.
Thank you, Horatio. There is another question. How can we define the true value in a drug product, since there can be a variation by 1% in values? I'm not sure if that's easy to understand, but that is how the question was
Yeah,
put up here.
Not completely understand, but I will try to respond. So during the variability, during the manufacturing process, obviously there are variability. Not all the tablets are equal. And then probably during the validation of the manufacturing procedure, you will identify that probability. Now, during the validation, remember that what we are trying is to separate all the variabilities associated with the manufacturing and only focus in the variability of the method. Sometimes can be done, sometimes it's more difficult, but that is the idea. So independently of the variability during the manufacturing, which is captured during the process validation, you have to assess the variability of the method in a separate form. And obviously, when you define the ATP, you have to take into account the process variability, the variability of the manufacturing process.
Thank you. We have also a few questions that are going in the direction if this chapter will also apply to cell therapy products, to other biological products. So we have three questions here that are going into that direction. So is it more for small molecules, or is it thought to be used overall?
Yeah, I think the initial concepts were developed with a small molecule in mind. Now, this question is not common. Is mentioned all the time. What about other methods? And we indicate in the introduction of the chapter that this approach can be used for any analytical procedure, but the effort you have to do in method development will depend on what type of method is. For example, in a method that is not quantitative method, probably there is not too much interest in expanding in the variability of the method in an ID test, for example. It would be more appropriate to use in a quantitative test, but some elements can be used. Now, and that's also incorporated bio. Bio products can also be analyzed. Methods can be developed using this approach. However, you have to accept certain flexibility, because this is not chromatography. It's not a typical instrumental test. So the idea of 1220, rather than give you specific directions, is to provide the concept, and you can choose how to apply the concept in the appropriate way depending on the method. Unfortunately, I'm not familiar with the cell-based products, but I'm pretty sure that the concepts can be applied in some way. Obviously, the variability of this method is much higher than a normal small molecule method.
Thank you. One more question we have here is about sample performance. And if you can explain the same, it's going into the direction to say a method is performing well in life cycle, a control sample can be tested. But how to identify such a sample is consistent, such a control sample is consistent and can be used then as such? Do you have some advice for?
Well, I mean, obviously not, because you will use one control sample today, another control sample in the future. But you will apply for that control sample the replication strategy. So that will give you a more better assurance of the method, because if everything is okay, the variability of the method will remain stable. So your replication strategy will give you a result with a plus minus total analytical error, and that is the value that you will compare with the future. So keep in mind that is sometimes the ongoing performance verification will imply more than one injection in the case of a chromatographic procedure.
Thanks, Horatio. We have two more questions. Maybe we can get them through before we end the call, the webinar. One question here that was just coming in: how can we determine the accuracy and precision for ATP before we develop the analytical method?
Okay, that's a good question. For the method itself, for the statistical tool you can use for that, anything that is appropriate can be used. There is no restrictions there. But again, the idea is you have your acceptance criteria for the critical quality attribute, and you know that is so critical that you cannot allow to have more than I would say 5% of the results outside the specification when you have a result that is close to the specification. So based on that, you can identify what is the appropriate accuracy and precision. Again, I will mention a rule of thumb, like you have to be sure that the total analytical error is only 1/3 of the total specification. That will give you some kind of a assurance that the method is appropriate. But any other approach is suitable.
And the last question that is in here in our question and answers box, there were also questions coming to the chat box, but we are looking at the moment just on the question and answers box here. That's the last question in here. Does the analytical lifecycle approach and also Q14 also apply to techniques that are employed for identification, so spectroscopic identification, or we have also loss on drying here, mentioned sulfated ash and residue on ignition, which is the same.
Again, in order to apply this approach, you need to consider how critical is the test you are doing. Tests like loss on drying or other type of tests, identification tests, it may be critical, but probably you cannot apply. There's no too much value in applying all this concept in a method like that. What I explained about identification before is that even though the chapter says that any kind of method can be used, can use the total enhanced approach or part of it, it is important to indicate that this really have value in quantitative tests that can be used for identification.
Thank you, Horatio. I'm just looking now into the chat. There are a few more questions. However, there are questions like, what can we do to reduce the analytical bias of a method? I think that's probably a little bit too broad to answer right now here. The same is here to what things we need to take into consideration to calculate the analytical error. I'm not sure if we can answer questions like these today. Would you like to try, or shall we?
Well, I mean, the total analytical error is a unified parameter that put together accuracy and precision. One thing to mention is a USP chapter 1210, statistical tools for method validation, describe one example of how to determine the total analytical error with to estimate of the estimate of accuracy and precision. So you have an example in chapter 1210.
In 1210, thank you. That's a very good advice here. Yeah. Okay, and the last question. Well, two questions maybe still. The sampling procedure, should that be included into the method validation protocol?
Good question. Yes, the method analytical procedure start with the sampling. It's very common to see a more extreme case where the operator prepare a stock solution and do several dilutions of those solutions to test the accuracy or to test the precision. That's not the appropriate way to do it. The appropriate way to do it is start from the beginning, from the weighting or from the sampling, in order to be sure that the method is working. So the answer is yes.
Thanks. And the last question that we have here, if I see a trend in my analytical results over the years in the chromatographic parameters, how can I distinguish a trend is based on the analytical method, so that we have a trend in the analytical method? How can I distinguish that from the trend that could be coming from the manufacturing process and is based on that? I think with a control sample, that would be a step.
That we talk about using the control sample. And again, one detail, I know that in many cases people are not familiar with the control charts. The control charts has specific rules about when to consider a trend and all, what to do when you have six continued trending results, or et cetera, et cetera. So I will recommend to study a little bit more the use of any type of quality tool, because there are many.
Thank you. Thank you very much, Horatio. I think we are now at the end of the question and answer session, and I would like to give back to Jan for his final words, please.
Can I make one announcement? The chapter now is being considered for ballot. The ballot will be in June of this year. Yes, in June, and it will be available in the USP, I think, in November this year. And the other comment is that we are planning a workshop between the British Pharmacopoeia and the USP for the end of September. So there is no announcement yet, but you will see the announcement.
Thanks, Horatio. That's useful information. Absolutely. Jan, please.
All right. Well, thank you so much, Horacio. Thank you so much, Christian, for this very interesting webinar. This will be recorded and available to participants, so please stay tuned as well for that. We have two more announcements for our next webinars coming up. Tuesday, 1st of June, we will have USP General Chapter 1469 nitrosamine impurities, which is then going to be followed again on Tuesday, 15th June, the value of pharmacopoeial reference standards, which will be presented by Christian, who's here. Christian, do you just want to mention like a line or two, sentence or two about what it is that you're going to be talking about?
Yeah, we will mainly talk about some challenges that you need to keep in mind when you are working with secondary standards and setting up your in-house reference standards in comparison to the pharmacopoeial materials or to the overall primary standard. So how you can take the least risks in doing that way.
Excellent. All right. Please, if you participants, if you still have any questions, you can still send them to us by email at emea.usp.org. If you also go on our homepage on www.usp.org, we do have an events and training calendar on the upper right corner, the right tab. If you click on it, you will see our other events and webinars and educational courses that USP is offering to you. Thank you so much again, Horacio. Thank you so much again, Christian.
Thank you for putting this together.
Wonderful session. And we hope to see you soon. Please stay safe and please take care of yourselves. Thank you.
Thanks for participating. Bye-bye.
Bye.
Thank you. Bye-bye.